Okay, so today we're diving deep into the history of CRISPR,
Oh, CRISPR.
which I am very excited about.
It's a good one.
It is a good one. So listeners, buckle up
Yeah.
because we are talking about a bacterial defense system
Okay.
that turned into a gene editing marvel,
Right.
a story full of happy accidents,
Yeah.
aha moments,
Yeah.
and even a little bit of scientific rivalry.
It's true. The story of CRISPR it has all the makings of like a scientific thriller, you know?
It really does.
And I think understanding how it all unfolded can really help us appreciate just how big of a deal this technology is and what it might achieve.
Yeah, absolutely. So to kick things off, let's rewind way back to 1993.
Okay.
1993, picture this. Scientist by the name of Francisco Mojica
Well, wake up, yeah.
is digging through bacterial DNA.
Oh.
And he stumbles upon these strange repeating sequences that are like
Yeah.
nothing anyone has seen before.
Like, what is this?
Exactly. Now, DNA, as you probably know, listeners, is like the instruction manual for life.
Right.
But these repeating sequences, it was like finding a hidden paragraph in that manual written in a language that no one understood.
Totally. And for years, Mojica himself, he was baffled.
Yeah.
These sequences, which he later called CRISPR, seemed to be part of the DNA repair machinery, but something about them just didn't fit.
Yeah.
It's like finding a piece of a jigsaw puzzle, but you have no idea what the bigger picture is even supposed to be.
And that puzzle piece sat around for over a decade.
I know, right?
But then fast forward to 2005,
Okay.
Alexander Bulletin, while studying the bacteria we use to make yogurt,
Yogurt, yeah.
discovers Cas9, a protein that would turn out to be, we now know, crucial to CRISPR. But even then, its purpose remained a total mystery.
Right, so it's like scientists had found this powerful tool,
Yes.
but they were missing the instruction manual. Like, they knew that Cas9 could interact with DNA, but they didn't know exactly how or why.
It seems like everyone was kind of circling around the answer,
Yeah.
but they couldn't quite grasp it.
Right.
So when did everything finally click?
Well, that's where things get really, really interesting.
Okay.
Enter Eugene Koonin in 2006.
Okay.
Now, Koonin wasn't actually working with bacteria in a lab. He was analyzing protein groups using computers.
Okay.
And this is, I think, a perfect example of how seemingly unrelated fields can collide to spark a major breakthrough.
That's so cool. It's like scientific cross-training at its finest. Exactly.
So what was Koonin's big revelation?
Well, he proposed this revolutionary idea. What if CRISPR wasn't about repairing DNA at all?
Okay.
What if it was a bacterial immune system?
Wait, bacteria have an immune system.
Oh, yeah, you bet they do.
I never thought about that.
Yeah, and Koonin believed that those mysterious repeating sequences that Mojica had found
Right.
were actually snippets of viral DNA, like a bacterial most wanted poster of past invaders.
So like a bacterial vaccine card. That'd be
Precisely.
incredible.
But like any good hypothesis,
Yeah.
Koonin's idea, it needed proof.
Of course.
Someone had to show this bacterial immune system in action.
Okay, and who stepped up to the plate?
That would be Philip Horvath in 2007.
Okay.
Remember those yogurt-making bacteria that Bulletin was studying?
Yeah. Horvath infected these bacteria with viruses.
Okay.
And he discovered that the bacteria were indeed incorporating bits of the viral DNA into their own CRISPR sequences.
What?
And even more intriguing, when those same viruses attacked again,
Yeah.
the bacteria recognized them
No way!
and fought them off.
So the bacteria were actually remembering past encounters
Yes.
and using that knowledge to protect themselves.
It was amazing. The first experimental proof of CRISPR as an adaptive immune system.
That's incredible.
It was huge.
It really is. It's blowing my mind. And it's amazing how something as simple as yogurt bacteria
I know.
could unlock such a profound secret.
It's pretty cool.
But we're just getting started. All right, what happened next?
Well, just a year later, John van der Oost made another crucial discovery. CRISPR uses RNA as its guide to target viruses.
Okay.
Now, RNA is like DNA's messenger.
Okay.
It carries genetic information within the cell.
So we're starting to see how the pieces of this puzzle fit together.
Yes.
We've got the CRISPR DNA, which acts like a memory bank of past viral infections.
Right.
And now we have RNA acting as a guide.
Exactly. But what no one knew yet was just how powerful this system could be.
Right.
And more importantly, that it could be harnessed to edit genes, not just in bacteria, but in any organism, including us.
Wait, hold on. Are you saying what I think you're saying?
Yeah,
This bacterial defense system could be the key to editing our own DNA.
you're catching on quickly. But before scientists could even dream of editing human genes, they needed to unravel a few more mysteries. Yeah, they had all these pieces,
Yeah.
but still a lot of mystery there.
It's like they had this incredible power tool,
Right.
but they were still figuring out all the buttons and settings,
Exactly.
So what was the next big aha moment?
Oh, in 2008, Luciano Marafini
Okay.
and Eric Sontheimer made a discovery that would change everything.
Okay.
Up until that point,
Yeah.
scientists had just assumed that CRISPR, with its RNA guide,
Yeah.
was targeting other RNA molecules for destruction.
Gotcha.
But
Yeah.
Marafini and Sontheimer proved that wasn't the whole story.
Okay, I'm on the edge of my seat.
Yeah.
What did they find?
They discovered that CRISPR wasn't just targeting RNA.
Okay.
It was actually targeting DNA.
Whoa.
And not just any DNA.
Yeah.
It was targeting and cutting the DNA of the invading viruses with incredible precision.
Okay, that's a game changer.
Yeah.
We're talking about, like, the blueprint of life here.
Exactly.
And CRISPR was capable of precisely snipping those blueprints.
Yeah, this was huge.
Yeah.
I mean, this discovery sent shockwaves through the scientific community.
Like that.
Because it was like realizing that this bacterial immune system, it wasn't just a shield, it was like a precision scalpel,
Yeah.
And suddenly, the possibility of using CRISPR to edit the DNA of any organism,
Yeah.
including humans,
Yeah.
became very real.
This is where the story really starts to feel like a scientific thriller.
It does.
Everyone must have been scrambling to figure out how to harness this power.
Oh, yeah, big time. And in 2010, Sylvain Moineau,
Okay.
he zeroed in on Cas9,
Okay.
that intriguing protein we talked about earlier.
Right, right.
Moineau confirmed that Cas9 was the one that was responsible for making those precise cuts in the DNA.
Okay.
And even more importantly,
Yeah.
he showed that Cas9 could be programmed to cut DNA at specific locations.
Programmed.
Yes.
So it's like giving Cas9 a set of coordinates
Yes.
and saying, go here and make a cut.
Exactly. Think of it like a pair of molecular scissors
Okay.
that you can program to cut DNA exactly where you want it.
Oh, wow.
And this was monumental.
Yeah.
Because it meant that scientists could potentially use Cas9 to make targeted edits to any gene in any organism.
It's getting good. It's like that moment where scientists realized they were holding the keys to, like, a whole new world of possibilities.
Right?
But something tells me we're not done yet.
Not even close. In 2011, Emmanuelle Charpentier and her team uncovered a missing link, a molecule called TrackRNA.
TrackRNA. That sounds like something out of a science fiction novel.
Oh, no, right?
What is that?
It might sound complicated,
Yeah.
but it's actually pretty simple.
Okay.
Think of TrackRNA as like the GPS system for Cas9.
Okay.
It acts like a guide, leading Cas9 to the precise location on the DNA molecule that needs to be cut.
Okay, that makes more sense.
Yeah.
So we've got the CRISPR DNA, which remembers past viral infections.
Right.
We've got RNA, which acts as a guide.
Yes.
And now we have TrackRNA, which is like a GPS system to make sure that Cas9 cuts the DNA in the right spot.
It's like the ultimate gene editing toolkit, right?
It's like the ultimate gene editing toolkit.
And around the same time,
Okay.
another scientist, Virginis Sixnies,
60s?
was making incredible strides on kind of a different front.
Okay.
He managed to take the entire CRISPR-Cas9 system from one bacteria and transplant it into another,
Wow.
proving that it could function in a totally different organism.
So it's like taking the engine out of one car, putting it into another,
Exactly.
and it actually worked.
Yeah. And it worked, which showed that this CRISPR-Cas9 system was incredibly adaptable.
Right.
It wasn't limited to just one type of bacteria. It had the potential to be used in a wide range of organisms.
This is all getting very exciting. Like we're talking about editing the very fabric of life itself.
I know, it's huge.
But I have to ask,
Yeah.
with all these incredible discoveries happening simultaneously,
Right.
who ultimately took the gold medal in this CRISPR race?
That's where the story, I think, takes another really fascinating turn.
Okay.
You see, around the same time that Sixnies was transplanting CRISPR systems,
Yeah.
Emmanuelle Charpentier teamed up with another prominent scientist, Jennifer Doudna.
Two brilliant scientists joining forces.
I know.
Okay.
And together, they published a landmark paper in 2012.
This has got to be big.
It was a pivotal moment for sure,
Yeah.
Doudna and Charpentier.
Yeah.
They really meticulously dissected the CRISPR-Cas9 system,
Okay.
revealing the precise roles of each component, and crucially outlining how it could be programmed to target and cut specific DNA sequences.
So they basically wrote the instruction manual.
Yes. It was like they wrote the instruction manual for CRISPR-based gene editing.
So this was it, the big breakthrough everyone had been waiting for.
It certainly seemed that way, but
Wow.
science is rarely a neat and tidy race with a clear winner.
Right.
Just as Doudna and Charpentier's groundbreaking paper was making waves,
Yeah.
another scientist, Feng Zhang, was
Wow.
making his own history-making discovery.
Oh no, a plot twist. This is getting too good. What did Zhang discover, and how did his work change the trajectory of, like, the CRISPR revolution?
Yeah.
Okay, I need to know.
Yeah.
What did Feng Zhang bring to the table that took CRISPR to, like, the next level?
Well, remember how Doudna and Charpentier had essentially created the instruction manual for CRISPR-based gene editing?
Yeah.
Zhang took those instructions and built something incredible
Okay.
in January of 2013,
Okay.
just a few months after Doudna and Charpentier's publication.
Oh.
He demonstrated for the first time that CRISPR-Cas9 could be used to edit the genes of human and mouse cells in a lab setting.
Wow, so he took this theoretical tool
Yeah.
and made it a reality.
Exactly. His team showed that you could introduce CRISPR-Cas9 into living mammalian cells,
Okay.
and it would target and cut specific genes, paving the way for targeted gene editing in more complex organisms.
This is huge. We're
Yeah.
talking about potentially curing genetic diseases,
Yeah,
engineering crops that are resistant to disease,
absolutely.
so much more. It's like science fiction has become science fact.
It really is, and it all happened so
That
fast.
really did.
In just a few decades, we've gone from these mysterious repeating sequences in bacteria to a tool with the potential to, like, reshape the world as we know it.
It's really remarkable when you think about it. So where do we go from here? What does the future hold for CRISPR?
That's the million-dollar question. Currently, scientists are working on refining CRISPR technology,
Okay.
making it even more precise and efficient.
Of course.
Imagine minimizing unintended edits, making it even safer and more reliable for use in humans.
Yeah, that makes sense. We want to make sure that we're using this powerful tool responsibly, right?
Absolutely.
But besides refining the technology itself, what are some of the most promising applications of CRISPR?
Oh, the possibilities are truly mind-boggling. Like, we could potentially cure genetic diseases that are currently untreatable.
Yeah.
Conditions like cystic fibrosis, Huntington's disease, sickle cell anemia,
Right.
maybe even some types of cancer.
It's incredible to think about the potential to alleviate so much suffering.
It is. It's huge.
Yeah.
And it goes beyond just treating diseases. Imagine engineering crops that are resistant to pests or diseases,
Right.
reducing our reliance on pesticides, increasing food security,
Wow.
or using CRISPR to develop new diagnostic tools that can detect diseases earlier and more accurately.
It sounds like CRISPR has the potential to revolutionize not just medicine,
Right.
but agriculture, energy production, environmental science.
It's crazy.
The list goes on. And we've covered a lot of ground in this deep dive.
Yeah, we really have.
But if there's one thing I want you to take away from today's episode, listeners, it's this. CRISPR's story is a testament to the power of scientific curiosity, collaboration,
Oh, yeah.
and perseverance.
Huge.
It reminds us that even the most groundbreaking discoveries often emerge from unexpected places, and that the pursuit of knowledge can lead to truly remarkable innovations.
I couldn't agree more. And it makes you wonder, what other scientific breakthroughs are just around the corner?
Yeah.
What will be the next CRISPR? What will be the next scientific revolution that, that captures our imaginations and changes the world?
That's a great question to ponder. And on that note, we'll wrap up this deep dive into the history of CRISPR.
I've enjoyed it.
Me too. We hope you enjoyed this journey through the world of gene editing as much as we did.